So yeah, so let's start. Hi everyone. I don't think we need to reintroduce in length the objective of the workshop, but just to say, for those who could not attend all the session, that everything has been recorded, is available on our website, and will be available for a few months at least. So and we'll see. This is part of the discussion to know what we're going to do with all this material. So today's session is a bit different, both in format and in content. Our objective is really to revisit some of the key points that have been raised during the sessions. So including calibration, standardization, bioeffects, monitoring, blah, blah, blah. And we go over this. And we plan to structure the session around 3 parts. Let me try to share my screen quickly. So this is what we had in mind. So our first part to go over the first 5 topics discussed in the 5 first sessions of the workshop, with about 20 minutes per session. We have a list of questions that were sent by the organizing committee to launch the discussion, and then it will be open discussions. We will take a break, maybe a first break in 1 hour. A second break in around noon. Second part of the session will be a discussion of partnerships with the IEC, FDA, and so on. And then we will discuss the next steps. The last part of the session will be a discussion around the next steps. This is again, just a summary of everything we discussed. So starting, the idea would be to create a framework for the discussion. So I'm looking at my chart. Perfect. Okay, super. So starting with the cavitation bubble detection, localization mapping, the threshold, the dose, the influence of cavitation agents. And then really the question, and maybe that will be for later, how can we relate all of these to bioeffects interaction? How can these bioeffects be related to therapeutic endpoints? And how can this inform on the clinical outcomes? So this is the road we basically trying to define. So for each of the question or topics, let's say, we would like to try to get a summary at the end of this session on the agreement on the existing knowledge, the knowledge gaps, and try to identify what are the opportunities and the challenges. This is what we want to do. So before we start, just a quick one about the logistics. As you realize, it's a giant open Zoom meeting. So we'll ask you to stay muted if you don't talk. Use the raise your hand signal if you want to share your thoughts. You can also send a message in the chat if we don't see you, or if we are too late to give you the mic. And just the last one before we start of acknowledgements. Again, just wanted to acknowledge the support of the AIUM and FIRST Foundation in organizing this event, both logistically and financially. And we wanted to share a warm thank you, especially to Emily Whipple. She's here at the beginning of this meeting from the FIRST Foundation, who really was instrumental in managing all the logistics for the workshop. And thanks to the organizing committee for their commitment, efforts, contribution, and it was a great journey. And of course, the speaker, panelists, and moderators, and all the audience for sharing and participating. And so now, maybe I will let Brian guide the first discussion on the session number 1, Brian.
So the idea that we had was to spend a few minutes talking about each of these topics. We'd asked the moderators to send us some additional questions, kind of the ones that are what they thought were the salient important questions to be addressed initially by the group. And as Fred indicated, then we'll step into kind of getting feedback from everyone on that 4-quadrant matrix of information that we might want to have related to that topic. So the first topic then is cavitation detection equipment, and there was a lot of very interesting discussion during that session, particularly with regard to sources and calibrations. The question that was raised by the moderators is, with the availability of a standardized point source to calibrate single element and phased array or array-based cavitation detectors, help with comparing results obtained across laboratories for cavitation-based therapies. So with that, I'll open up the discussion and hear anyone's perspective on the need for such point source calibrations.
Well, I will make a start. So some of the motivations behind that suggestion were twofold. One, to enable better comparison of the sensitivities and frequency responses of cavitation detectors across labs, even those labs that are expert. But secondly, which I think is also a huge intent of this workshop and this endeavor, was to also enable groups that perhaps use cavitation and ultrasound as a tool, but don't have expert in-house expertise, to have an easy way of informing the community and submitting results for publication what the characteristics of the cavitation detector used was. And so overall, my feeling is that that would be an excellent endeavor if we could make it easy enough and if we could make it cost effective enough. One of the ideas we discussed was the possibility of producing such a point source potentially cheaply enough to even be a single-use disposable or a limited uses disposable, because one of the issues, of course, will be calibration of that very point source. And the second is to what extent a single point source could be used to serve different frequency ranges, to which the expert answer was most probably not. So very supportive of the idea. I think it would bridge a lot of these gaps we have identified. We just need to work hard as a community to make it really easy to use and really clear on what it is to be used for.
Okay. Other comments, other thoughts?
There was one that came up in the chat.
Yeah, I see it now from Sam Howard. So Fred, I saw your hand pop up.
Oh, okay. Yeah, it was and to follow on this discussion. I mean, do we how let's say, to what level do we need calibration? Let's say, let's write it like that. To what extent do we need calibration? You think?
So I think the question of detector sensitivity is probably the critical thing in my mind. And without being able to know what that detector sensitivity is, it's very difficult then to understand whether or not, when we get to the point of looking at cavitation dose or other types of metrics, whether or not a given system is sensitive enough to be able to perform at the same level as other systems have done. And so that seems pretty critical to me. I see Kevin Hayworth's hand is up. Kevin?
Yeah, I think I would just add to that. I think, so I agree with everything said. I think it would be helpful to have something on the hardware side. I think we also, though, need some standardization on the signal processing, right? So once we digitize that signal, how do we make sure that people are treating that appropriately also? There's a lot of play in how you can run FFTs and all of those sorts of things.
And one thing I would ask, I mean, for folks that are on the call, if there are things that you think are important to capture, and I want to make sure that we're mindful, although we're recording the session, if you could transmit those to me or to Fred, I think that would be very, very helpful in creating a summary of the discussion. So if you detect things in this, no pun intended, then please convey those to us. Other thoughts?
I thought Brian, Sam's suggestion on estimating received sensitivity from a pulse echo measurement would be interesting to pick up, because that speaks directly to Kevin's point. I mean, all ultrasound labs have a pulsar receiver. All non-ultrasound expert labs don't necessarily have a pulsar receiver. And in my view, that speaks even more loudly to the necessity of us not just having the point source, but also perhaps an agreed and an openly shared script and pathway for the entire signal processing path, to Kevin's point. So if we could actually not just do the detector, but also the back end of the detector with a very clear manual on how to perform and report this measurement, that would help. There's also a point from Costas and from Kevin I'm seeing pop up, so I'll hand over to you.
Yeah, I see Costas' hand is up too.
Yeah, I think just was the last comment by Constantine just captured what I want to mention is that calibrated point source. I think it's quite important, and being able to have this point source that will be available to everybody. But how we're gonna do the measurement, I think, is also critical and has become very clear from the first day from the first session of this workshop. So I think those goes down to some details that need to be added on how this will be performed, and whether a single measurement or a spatial measurement will be needed. But certainly I think that's pretty much captures what was the main idea and what were the main ideas and main topics covered in the first session. So we certainly need to be in position to at least have an idea about, when two labs report some measurements, whether these measurements have been performed with systems that have comparable sensitivity.
Okay. So being somewhat mindful of the time, which is the moderator's job, it looks like we've hit a lot on probably what was the second question that's sitting on the screen right now, which is how can we best establish standardization or practices for processing, quantification, and reporting of results. Would it make sense at this stage to form an application-specific working groups? That's something that we haven't talked about. So do we think that's probably something we should be looking at, is application-specific working groups that are related to these types of quantifications? Kevin?
So I think the answer is both yes and no. On the no side, I think there's a lot of the processing that can go on that's just purely signal processing, right? Making sure we handle the signals appropriately and we have good sensitivity information on there. But then how you then convert that signal into some sort of metric of dose, and I'm sure we'll get into that later, right? But really talking, I guess, about how we quantify that signal, I think, yes, then that will require application-specific working groups. So I think the first part of the processing, we don't necessarily need that, but then on the second part we do, would be my thought.
Yeah, that seems to make some sense. Fred?
Sorry, checking with you. Yeah, so in terms of gaps of knowledge or state of knowledge, in terms of this calibration, even software or hardware, do we know exactly what we want to measure, or how we want to process the signal? I mean, what we want to extract from the measurements. I'm not talking about metrics that would be related to bioeffects, but do we know exactly, is there a consensus of we should be measuring? If we insanify at, let's say, I don't know, 2 megahertz, we should be looking at a range of frequency from such to such, or I mean, is there a consensus, or is there still some gaps in knowledge in terms of just assessing or measuring the cavitation noise?
Constantine?
For me, the key metric that we want to get out of a detector is acoustic power spectral density. So in other words, what we want to know is what the sensitivity of the detector is over its usable frequency range. Because if we have that metric even for different detectors, we can then in some sense compare, at least over the frequency region of overlap, the acoustic emissions detected. And I think we need to come up with two scenarios which came up in session 1 and session 2. A simpler way of doing this for single element detectors, whether they are single element focused PCDs or even hydrophones, which some people are using as cavitation detectors. And a second methodology, which Christy alluded to in a number of her comments, for arrays. So with increasing use of PAM, we also need to make it easy for people to calibrate arrays on receive. And this calibrated point source and signal processing path could help with both. And I really think the key metric is acoustic power spectral density.
Okay, so I'm going to stop sharing my screen. I think probably Fred has figured out how to put up his slideshow with the 4-corner matrix, and then I think Kevin, you had your hand up.
I did. I guess I would. I'll ask this as a clarification point from Constantine on that. I generally agree with that, but I think, is this the power spectral density incident on the receiver, right? Or is this trying to propagate it back into a measurement in the field? Even if that's, you know, with just a single element PCD, right? You could try and do that. So I think we need to figure out, I think the former is much, much easier. The latter is probably much more relevant to a bioeffect.
You're absolutely right to pick up on this point, and it's a subtle but very important one. I think in the first instance, I'd be very pleased if we could start by achieving the former, i.e. what happens on the surface of the receiver. There is no question that for some more advanced investigations, you know, phase information might become extremely important. And being able to propagate it forward into the point of origin would also be highly desirable. But I suspect the latter will be even more difficult to propagate across laboratories unless a really clever person, perhaps the person, that one who asked the question, can find a really easy way of doing it.
I appreciate that, Constantine. Let's turf it to somebody. So with the matrix that you see up on the screen, it sounds like we've got a pretty good bit of existing knowledge for the calibration problem. We kind of know what the problem is. It sounds like some of the knowledge gaps are the utility of the point source and maybe this question of the back propagation back into the field. Other knowledge gaps as we wrap up this component, we'll spend the next 5 minutes kind of working on this matrix. Costas?
I just wanted to add that while there is going to be a lot of discussion about what the y-axis should be of this power spectral method, I also want to mention that it will be desirable, if possible, to have background data in the absence of microbubble activity. And I think combining those two might also create opportunities for self-calibration, or might allow to relax the needs for quantifying the acoustic emissions, where there is a big topic, and I think we'll have many and extended discussions about that.
Yeah, background signal as a reference is an interesting proposition, and something that we might be able to combine with the KIBA activities for the pulse echo quantification, what's called the PICUS group at KIBA that's trying to get that. Michael?
Yeah, I think for array systems that are trying to image over a large field of view, receiver directivity as a function of depth and source position becomes a challenge, mostly because you just have to make a lot of measurements. And if we're trying to gear this solution that we're coming up with to people that are not specialists and don't have all the sophisticated equipment that some specialist labs do have, my suggestion is that perhaps a way forward there is to recommend a smaller subset of measurements to be accompanied by a validated model that allows you to get a good handle on all the other things that might be going on. So a simple measurement plus some calculations to support estimations for the rest of the field. And I think that's just a matter of working out what is that model in order to make this technique overall more accessible to the community.
Well, and with that thought in mind, I'd refer folks to the chat and the comment that Sam Howard made about whether or not, at what level are existing equipment and methodologies available. And I think that falls under the paradigm of outlining what our existing knowledge is and what then the need is for different types of equipment development. And that, I would say, follows then under potential opportunities, particularly for the companies involved in developing equipment, both for the production and the detection of ultrasound fields and their quantification. And I know that there's a lot of standards that are out in the field that we can leverage, and a lot of knowledge that we can leverage out in the field related to that. And I think we've hit on a lot of the challenges. Are there any challenges that we haven't hit, just so that we enumerate those, and any other items that kind of wrap up this particular area?
I mean, I just want to comment that we talk about calibration and quantification, right? So quantification remains a big challenge. I mean, we have to acknowledge that even if we have a perfectly calibrated point source and a perfectly calibrated system, the units that we'll have will not necessarily reflect the microbubble activity or what we think as a tension exerted by the microbubble. So I think the quantification is what this means. The y-axis is a big challenge.
Okay.
The only other thing I would suggest as an answer to your question, Brian, is after we put this together, we also need to ask ourselves as a community, does following this procedure ensure that if you get two experiments with the exact same calibrated point source and you get the exact same answer, but you observe something different, why could that be? So we need to provide, no method will ever be perfect, and we need to get our heads together to figure out why the reasons for imperfections of that methodology might be, to get people to either mitigate them or at least acknowledge them.
I think that's a huge point, Constantine, and a lot of what the KIBA activities kind of are meant to drive toward, which is to figure out the sources of bias and variance in measurements in a productive way, to be able to drive those toward resolution. In other words, what can we really expect in these measurements to be a level of repeatability, reproducibility, and understanding the sources of those bias and variance. In the end, you can't eliminate them. The only thing that you can do is understand them and what their sources are, and try and mitigate those sources. So we're kind of at the end of our time for this particular topic. So this is going to be an ongoing discussion. So it's very important that we capture these things and then figure out, and we'll talk about that later, what our next steps are. So Fred, do you want to take the next topic?
Yeah, and I think so it was about cavitation localization and mapping, but I think some of the questions that we received to start the discussions are really linked to what we just discussed. So the first one was, so how do we establish calibration approaches that can be implemented by groups that do not have a focus in ultrasound metrology? So I think we already discussed some of these, but do you have any other thoughts? So one interesting idea is, of course, to combine some type of measurements with some type of modelization. But still, that could be a little bit heavy for groups that don't have a focus in ultrasound metrology. So is there any other ideas, propositions for that? Yes, Kevin?
I like Mike's idea of, trying to combine simpler measurements with modeling, as you just said. I wonder, in the context of doing this, if setting things up kind of how, some of the guideline statements in medicine work, where, there's a preferred approach and then a secondary approach if the first one doesn't work. I mean, that's a little bit more with, patient risk factors, but kind of thinking analogously, if there's something like a tier 1 approach, which would have, all the complexities of the measurements for the labs that can do that, and a tier 2 approach that would be, a simpler, make these simpler measurements and combine it with this modeling. And then, I don't know, maybe there's multiple tiers within that. But rather than thinking about this from a only one option, maybe it's a ranked order of options.
Yeah, I think that's very interesting idea. Yeah. Is Gail on the call? I don't see. I can see everybody, all the participants at once.
I don't believe she's Frederic.
Is not okay. Okay. So yeah, this is the idea. I mean, we so Gail published a paper 10 years ago, the guidelines for reporting therapeutic ultrasound treatments parameters, and we are working on a updated, let's say, version of these guidelines, where we want to apply exactly this idea, Kevin, of having different level of characterization based on the abilities and equipment of the group. So I think that's a good idea to keep in mind. Are there any other comments on this question of how to make it really accessible to non-metrology groups? Looking at the chat.
Yeah, so it's the point you just made, Fred, about Gail's excellent paper on standardizing transmit criteria for transducers. So we could, of course, apply it in reverse on receive criteria for PCDs. So you'd still need to know the material, the focal length, the electromechanical efficiency. This may be made redundant by a standardized calibration source. So and it's not a leading question. It's an interesting question whether, if we have a really good standardized source, we actually need both or we don't. Because arguably a well-reported calibration from a standardized calibration source should eliminate a lot of the differences between detectors of different focal lengths or different sensitivities. But I think that is something we should definitely discuss as part of the expert paper and the subsequent discussion.
So no other thoughts on this? I'm looking at the chat.
I wonder, Brian, I don't know if you could comment on this as kind of a follow-up to Constantine's, but the thing you were talking about with Kiba, right? We're trying to understand, biases between groups. So if we have, a perfect, way to calibrate, I agree with Constantine's comment, but we know we're going to have biases and that sort of thing. in which case then being able to reconcile those differences, it would be beneficial to have more information about equipment setup and stuff like that as kind of a companion paper to what Gail published. So has your experience been with Kiba that things are wide enough that we do need quite a bit of information still provided? Or is it, you know, if you follow these standardized procedures, that's good enough and you can skip reporting a lot of things?
I hate to say that it varies. I'll use your yes and no, just like that you did before. The only thing that we know is that following some level of standardized procedure in terms of how the instrumentation is handled and how the operators actually run the instrumentation, then you can achieve a certain level of bias and variability in the operations of those. The best probably information that we have on the ultrasound side right now in terms of as the furthest along in the Kiba process has to do with shear wave. And in the end, what was discovered is that because the systems were already operating in a certain way and measuring their shear wave using their own methodologies, we had to find a way of being able to understand each of those systems and how they relate to each other. It's much better if you start probably at the level where we are right now, which is we create a standardization where people are operating systems the same way. They're using the same methodologies, where you don't have that level of variance that exists because the methodologies are different. The key in some cases is you don't want to stifle creativity. And that's where things get to be a little dicey, because yes, standardization is good, but also innovation is good. So we have to be mindful that if there are new and interesting ways of being able to do it, we can standardize on certain methods, but we need to be mindful that a better method may be discovered, in which case we need to be able to accommodate that. Does that help? Okay.
Which can transition to the next question, maybe that we already a little bit discussed, which is how to validate this standardization calibration approaches. What's
I see Christy's hands up.
Yeah, I see. Yeah, sorry, Christy, go ahead.
Hi. So, we've made an attempt at calibration in our lab, and even having a printed circuit board that you can plug into the boot of the array to tap off all the voltages, that doesn't actually change the impedance, is not straightforward. And I think there could be some creativity in that area. We have these standardized boots for most of the array transducers, let alone matrix, right? But encouraging that creativity and standardization, I think, would be a first step. You kind of need to be able to measure the sensitivity of each element. And right now, most of those arrays are closed, right? You can't just tap in. So I think that would be a first step.
So do you think we need phantoms in addition to measurements of this sensitivity, let's say, of the elements? Will this be a requirement to have some kind of cavitation, phantom that we need to use to validate these calibration approaches?
I do think that you need standardized scatterers. That's the next question is, once you can measure those voltages, now what are you scattering off of? Bubbles? Are you scattering off point targets? What is that going to be? So that's something else we need to talk about. And I'm sorry, I joined late. I got my booster today. So you may have already talked about these things.
No, we didn't. I mean,
okay.
I see that James, please go on.
Hello. Hi, everyone. So what we do in our lab is we use shock waves from microbubbles, and they do a fairly good job at calibrating the sensors because they're very small and they contain a very, very broad frequency range. And so that's what we use. We also saw papers where they're focusing sound using calibrated emitters, and they do quite well with focused PCDs. So those are two possible routes that probably a lot of you are already aware of. But the overall topic, the whole idea of calibrating your transducers is really, really a good thing that we should all try to go for, but it's hugely problematic for many of the reasons that you guys have mentioned over the workshops over the last few weeks. My main concern with that is spatial averaging on your sensor. And if you move your point source a little bit left or right, you're just going to get lots of spatial averaging. So I think for me, the most important thing is to just make sure that everyone's reporting their full setup and the arrangement, and that needs to be very thoroughly reported. And when I read some papers, sometimes that piece of information is not all there, and that would really help me interpret what exactly is being visible. Because if you just take the same sensor and you move it to another position, you'll get wildly different results from your PCD readout. So those are just my thoughts.
No, thanks. And the question of how to let's say standardize the reporting of equipment and method is a question that we will discuss a bit later.
Okay.
Sorry, I'm checking at the chat. Okay, so is Adam here? No, so Constantine, go ahead, please.
Thank you. So Christy, you mentioned this sort of breakout box and impedance issue on a couple of occasions, and I just wanted to try and understand it a little bit better. So clearly, we're trying to understand the sensitivity of each and every element on an array that is going to be used passively. Presumably, if we were to have access to a calibrated point source, that would sort of negate, and that point source was reasonably broadband, that would negate the need to have either a scatter or a shock wave generated by a bubble. I mean, all of these three things would effectively serve the purpose of producing a known broadband point source of known acoustic amplitude. So I'm not entirely sure why you would disconnect the array from the Bob connector rather than just receive the signal, the voltage trace, in the way you would normally receive it using your Verasonics or other data acquisition device. Do you want to be using the exact same impedance as your detector would be using when used as a passive cavitation detector? I'm missing something in the point you're trying to make, and I'm just trying to understand it.
Adam, why don't you follow up? I see your hand raised.
Sure, yeah, happy to. So I think the point that Christy was trying to make is that we can have an independent measurement of the voltage or the impedance of the transducer elements in cases where we have a transducer where we can't readily access the voltages at the interface with any sort of detection system. So having a breakout board allows you to have your own electrical measurement scheme, and this could be some standardized scheme of between labs to be able to independently record that impedance of, say, an imaging probe or something while it's connected to the system, and also to be able to individually measure the voltages received on each channel for that same system when otherwise you wouldn't be able to make any sort of independent measurement of it.
So the purpose is to decouple the electromechanical response of the sensor from the electrical response of the DAC system.
Right, or in cases where you may not have access to all the details of how that signal is being processed by the system or by the hardware of the system, you can have a measurement where you do understand all the electrical aspects of it that are in between the transducer and the system.
That makes sense. Thank you. Thank you, Adam. Thank you, Christian. Understand now.
Okay,
I
thank
think
you.
there's a comment by Costas.
Yes, and then we move to the next topic.
Yeah, I think we're spot on now in where the direction is heading to, because again, going back to the notion of incremental measurements and standards, and as well as between imposing some standards and not like limiting creativity, I think that all this comes together when we talk about image reconstruction and beamforming methods. While processing the data from the single element might be much easier to standardize, I think how we reconstruct the acoustic fields is quite different across different labs, and there are many new methods that you can do so. So I think being able to potentially characterize one element of the array that will then be used for maybe some reconstruction algorithms will allow us to at least being able to compare across different methods and potentially different systems.
Yeah. Thanks, Costas. Adam, you want a last comment? Short.
Yeah, I just wanted to ask if there have been attempts to standardize between single element and array transducers in a way where you could use the beamforming of the array to approximate what you would get from a single element PCD. I wonder if there's a way that we could have a single or a way of processing array data that you could beamform it and report it that would be equivalent in terms of like the acoustic power spectral density on the array to a single PCD for comparison purposes.
That's an interesting proposition, something that we probably should think about.
Yeah.
So before we jump to the next topic, just briefly, if you slide down one slide, Fred, the question is, with this matrix, are there any final comments then about, I would say, knowledge gaps, challenges, and opportunities? We've talked a lot about existing knowledge. Are there things that we haven't captured in this conversation, thinking specifically about cavitation localization and mapping, that we need to address? Kevin.
I'd like to see on that reconstruction and beamforming, I'd like to see more of the actual scripts published for it, and not just having the the algorithms in their mathematical form, because I think again there's always a lot of detail in how that works with the reconstruction that will inevitably get left out, and I don't think we can report it just by writing it in the paper, because papers will get too long. So including a supplemental file of here's our beamforming algorithm would be great.
And I think that was actually an opportunity that was identified in previous discussions. So that's really good to bring that back up. Michael? You're muted if you're talking.
Thank you. Something that I think would go very nicely with what Kevin just pointed out is that along with documentation of what the beamforming and processing algorithms are, is to actually display and quantify a point spread function response in a certain frequency band, so that we know what the output of that thing is supposed to actually provide. That's what ultimately we visualize with these maps and with localization, and knowing what the size of these point spread functions is will tell us a lot whether we're really localizing very well or not.
Okay. So basically, documentation of some sort of independent documentation of the point source characteristics.
Yeah,
Okay,
I think
so
that's fair. Yeah, just a kind of a standard problem that doesn't require an experiment to solve.
Which once again I think would be addressed, Mike, correct me if I'm wrong, by the standardized point source, right? So if we had a broadband point hydrophone we could put in front of these arrays as a standard transmitter, that would go a long way towards answering that. Right?
Sure.
Yeah, and that's something that we can look at in the future, because that's been a common methodology for doing round robin type laboratory assessments. How well do people, if you share a point source and you send it around, then how well are people actually able to make common measurements? I know Costa and Kevin, you both have your hands up. It's like really fast. Costas.
Sure. I don't know if the comment is on opportunities and challenges, but I think with the passive, with the imaging arrays, because all the calibration happens in free field, I think the imaging arrays might provide the opportunity to account for different tissues and losses that might take place when we do experiments under more realistic conditions.
Now, that may be a great thing to cover in this next session that's coming up. Kevin.
Yeah, very quickly, I mean, as the Bubble Lab, amongst others, have shown, right, how algorithms handle interference between multiple sources is also a very critical component. So while I'm in favor of displaying a point spread function, and I like that idea as a first step, I think we need to make sure that it's very clear that they can also act very differently when there are multiple sources, especially for these data adaptive algorithms.
Okay. So that may fall somewhat under the challenges banner of making sure that we figure out how to deal with that. So we might actually need to put two sources in and see how those interfere with one another and how that's managed. So we need to move on to the next bit. So if Fred can advance it. So the third session, and then after this we'll take a very brief break. Was the cavitation thresholds and dose. And so while I understand all of the calibrations that have to do with everything that we just talked about, I'd like to spend a few minutes concentrating on things that are relevant in terms of thresholds and dose, because that takes us to that next level of understanding. So the first question, you see the three questions that are here, but possible metrics of dose, I think we've heard a little bit about that. Any additional thoughts, and particularly with regard to application areas, what could be possibly generalized? James.
So I'm not sure if this is the right forum to raise this, but the whole idea of the inertial cavitation dose, I see a lot of papers looking at broadband noise, which is what I was looking at in the past, but is that may be sufficient to say that there's inertial cavitation, but if there is no broadband noise detected by your sensor, there could still be inertial cavitation within the focal volume. And that might not be detectable because of spatial averaging effects. The fact that the transducer, the sensor is not calibrated, not just by the magnitude, but more importantly by the phase. And so just this definition of inertial cavitation threshold, the pressure threshold, I'd like to push back a little bit on that and say maybe we're not detecting that threshold with our sensors because it's not capable of doing that. I don't know what people's thoughts are on that.
I think you raise a very good point. I mean, detector sensitivity for me has been a long-standing issue, and especially if there's phase cancellation that's happening as a consequence of what's happening in the field. Constantine.
Yeah, so it's a very important and a very fair point, as you say, Brian. I mean, it's first of all, it's a really big reason why several of us believe some form of calibration is necessary, because at the very least, if one lab says there is no inertial cavitation in this set of parameters and another lab says there is, we need to be able to check whether there is significant difference in sensitivity of the receiving sensor. But I think when one is doing cavitation threshold measurements, going from a low pressure to a high pressure, you typically see one of two behaviors. If you're looking, let's imagine specifically for broadband noise emissions, you either get to a point where you see a broadband noise signature slowly arising from noise. And so at the first point where it emerges from noise, it is just barely detectable. And to me, that would be an indication of a very weakly conditioned system, because it probably means the real threshold is somewhere buried in the noise. In a well-conditioned system with a very high sensitivity, as you go from left to right, low amplitudes to high amplitudes, suddenly something dramatic happens where you go from nothing to being 6 standard deviations above your noise floor. And there, I think there is an argument for saying that's a true threshold. And I think part of the metrics should be, I'm not trying to answer the question because I think the question is still very open-ended. I think part of the question should be getting some sort of consensus of what is defined as a threshold. So what sudden change on the x-axis and on the y-axis do you need simultaneously relative to your noise floor to believe you've hit it?
Constantine, that's such a valuable point. That detector sensitivity and the level at which you can detect cavitation and whether or not it's 1, 2, 3 standard deviations above the noise floor is a pretty critical point, I think. As well as, you know, you can do calibrations for a particular, you know, standard particle, so to speak, and you would really want to know some methodology for being able to say, does that apply then in the context of bubble activity? In other words, cavitation noise and the associated differences in the type of noise, depending on whether or not it's inertial or not, non-inertial type cavitation is pretty critical, I think. I don't think all the answers come from doing scattering from solid particles where there isn't the nuances associated with bubble activity.
Because she's in a place with bad broadband, I'd also like to voice a point Eleanor Stride is making. I do think, though, that the different mechanisms underpinning bioeffects cannot be ignored. The energy emitted may be a very good indicator for thermal effects, but not necessarily for mechanical, for which peak pressure emitted may be more relevant as indicative of wall acceleration. And this actually joins the first bullet point that's on the slide, where if you notice, there's both mention of energy power density, but also peak power and time averaged energy. So it'd be interesting to hear from the audience which of these metrics that either relate to the peak amplitude of acoustic emissions or to the time averaged amplitude of acoustic emissions may be more relevant to some of the bioeffects they're working on, because I guess that's where those conversations started.
Kostas?
I think with respect to the point that Constantine brings up, I think we kind of like have split microbubble activity or cavitation in stable and inertial. And in that respect, I think stable cavitation should be measured in terms of average energy, and inertial should be referenced in terms of occurrences, in addition to amplitude that the signal has, in terms of the amplitude that the collapse has generated. So I think with inertial cavitation, I think it's also important to count the occurrences, because if we go for averaging in the inertial cavitation, it might be lost because it might happen a couple of occurrences out of like a very long sonication.
And I know, Christy, you were advocating for a particular cavitation dose.
Yeah, I just tossed it out there really so that it would start some conversation, because, you know, you really got to talk units at some point. What exactly do we mean? And we're in a transition too, where we don't have calibrated receivers yet. So whatever we do, I think it should be reported in certain units that can then easily be converted once these arrays are calibrated in some standardized way. So you don't lose all of the bioeffect studies that are reporting certain type of bubble activity, for instance. You can still compare your results with things that have happened in the past. And I'm assuming not everybody can calibrate their arrays just quite yet.
So that's a great thought. I think you would also need, however, to capture even electrically the frequency dependence.
Yeah.
So it needs to be some sort of spectral density. In other words, you know, there needs to be a function of frequency there, because when you get your calibration, it will be as a function of frequency. And Christy, to your other point, do you think we should always be reporting peak and time average acoustic power for the very reason you state, that is very bioeffect dependent?
Yeah, perhaps, or at least save the data so you can go back and reanalyze it if it becomes necessary. But I think it really depends on the bioeffect. I mean, if you don't expect it to matter, if the peaks are very transient and they're not going to contribute overall to the specific bioeffect, then, you know, they're kind of irrelevant. And I wouldn't want to force the, you know, it's very onerous to start reporting all this stuff. Then you get people that don't comply because it's just too difficult. I really think it depends on the bioeffect.
Yeah, I think the analogy of kind of with regard to safety indices for diagnostic ultrasound, one is probably more related to time average and the other more related to peak. And so if in the field we were able to decide on a couple of metrics that would work, that's probably something that could be manageable. But I think there has to be a scientific basis for their selection. And that may take a little bit more work to see whether or not they work in a given application or across applications. And that may be a little bit of a challenge. Kevin, final word before we progress to the matrix?
Yeah, sure. And I guess this kind of actually fits in with the matrix, so we can use that as our transition there. I think one of the things we might consider is, do we want to build something equivalent to a biobank, but instead for, you know, a repository where people can deposit some bioeffect data, the emission data that they have, right, and build up abilities for other people to go in and mine that, right, and combine some of those data sets, you know, just like genomics folks do, or, you know, any of that sort of thing.
I really like that idea. I think at some level, being able to create a repository, a cavitation repository, is something that would allow the field to run different algorithms on different types of data and those types of things. I think that's a real opportunity for being able to see whether or not a given dose scheme actually works for a given application. And that may be a methodology for moving the field relatively quickly forward. So in the last couple of minutes that we have, I remind you of this matrix. We talked a lot about the existing knowledge. We've hit on a few kind of knowledge gaps, such as maybe we need to define a cavitation threshold, or how that actually could be made with regard to detector sensitivity. Are there other opportunities, challenges, knowledge gaps that we haven't covered in the course of this conversation that we need to grab at this point? Okay, so based on the schedule, we're supposed to restart at 11:10, so we're going to take about a 10-minute break. So 11:10, if everybody could rejoin us, then we'll pick up with cavitation agents.
Let's resume and move on to the topic of the session number 4, which was about cavitation agents. So I think the first question was about what type of agents should we be using in different, and this could be, of course, application dependent, but should we use existing approved imaging agents, microbubbles, or develop specific ones? So does someone wants to start the discussion on that? So maybe a reminder that we had during the session different, of course, viewpoints, and again that could be really application specifics. It seems, I mean, one of the comment that was made, for example, for BBB opening was that the problem being already so complex, and the comparison of different bubbles didn't bring any strong differences in terms of efficacy of BBB opening. Maybe the best way to proceed was to try to work with the company that are developing already approved imaging agents for therapeutic use, rather than going through the all the others of developing specific ones. But again, this could be application specifics. So does anyone wants to start the discussion on these specific points? So I see Christy comment that limitation due of with the gas containing agents that could be coming from regulatory orders. Christy, do you want to comment further?
I just mean, drug versus device. The fact that you might have to go through both, or which is it, and it changes. And I mean, people that invest that kind of money really want to know an expected pathway, and not it to switch, or, and especially not to have to go through two pathways.
Yeah, so there was an interesting comment that was made by Keith Ware last week, if I'm correct, about the fact that the FDA, but maybe Brian, you can comment in more detailed manner, but basically the FDA was considering revising its strategies for bubbles from and moving toward the therapeutic device categorization. Brian, do you want to expand a bit on that?
And I think Keith is on the call as well.
Oh, sure.
There's a court case that has kind of been an impetus for this consideration, and it isn't with regard to therapeutic devices. It's more generally whether or not contrast agents in the diagnostic space should be considered either a drug or a device. And so it has, the ramifications are more far-reaching than just the application of contrast agents, microbubble contrast agents, toward therapeutic application. So Keith, comment?
Yeah, that's right, Brian. Since 1997, FDA has regulated all imaging contrast agents as drugs, even though contrast agents meet the legal definition of both drugs and devices. FDA has felt that it had the discretion to choose either regulatory pathway, depending on which one it thought was appropriate. And in response to complaints back in the 90s from industry that contrast agents were going through both paths, so there were concerns about inconsistent regulation, FDA in 1997 decided to do all contrast agent regulation as drugs in the Center for Device Evaluation and Research. But recently, a contrast agent manufacturer wanted their agent regulated as a device because they thought it would be to their advantage, because the laws regarding device regulation and drug regulation are different. This went to court, as Brian just said, went to a district court and a federal court of appeals, and the court ruled that FDA did not have that discretion that it assumed it had to regulate contrast agents either way, drugs or devices. And the court ruled that FDA must regulate a contrast agent as a device if it meets the legal definition of a device, which includes things like it does not achieve its primary intended purposes through chemical action within or on the body, and it's not dependent on being metabolized to achieve its primary intended purposes. So FDA is going through the process of examining all contrast agents and to find out which ones do meet the device definition. And there's a public comment period going on now. It's been extended until November 30th. And if people on this call want to make comments to the FDA about how they feel this process should be conducted, please contact Shubha Marivada or me, and we can tell you how to submit your comments. So the whole thing is a bit up in the air right now, but once the public comment period ends on November 30th, FDA will try to work out a solution that works out the best for the stakeholders while still being consistent with the court ruling and interferes in a minimal way with delivering healthcare to the patients. So that's where things stand right now. Sorry, I can't be more definitive.
Oh, thanks, Keith. That's very, I think, a very interesting comment. So going back to this question of so there are the regulatory aspects of using cavitation agents, and I think we covered the last question that is on the slide. But going back now to the first one, I think the one of the underlying question is finally what should be the ideal characteristics of cavitation agents for therapeutic applications. Again, could be application specifics, but are we satisfied with the existing macroballs, for example, that we have? Should we develop other agents? So again, do you have any comments? Go ahead, Konstantin.
So I'll try and kick things off. I think this is where we have an obligation to close the loop with some of the previous topics we discussed. One of the final comments made by Christy in the last section and by Eleanor is, is it peak power or peak amplitude or time averaged amplitude of the bubble oscillation that matters? Well, that is a crucial question, because if you can actually identify which bubble behavior drives your bioeffect, then and only then can you really answer, do current microbubbles do the job or don't they? So from a regulatory and a commercial perspective, if it is possible to achieve what one wants to achieve with an approved agent that has gone into human before, it makes complete sense to use it because it's much less painful and much faster to clinical impact. But if there are functional reasons why current contrast agents cannot achieve the intended mode of use, then we need to find both a new agent and a regulatory pathway to get it to patients. And a couple of examples from our own experience that spring to mind is, we started working on an application to inject microbubbles into the disc, the spinal disc. And it turned out that microbubbles simply couldn't survive the injection process against the back pressure. So you ended up with no nuclei at the other end. So there was no question. You needed something like the nanocups or the sonotran particles that were developed by Ultrasonics that could resist that back pressure. So if there is a functional requirement for a different agent, it needs to be identified, and it's a no-brainer that it needs to be developed and have a path to regulatory approval. If there isn't, go with what we have.
Yeah, so you agree. Yeah, that's a good point, of course. And in terms of agents, does anybody has any comments to make on this approach of moving to droplets, creating droplets from existing microbubbles? Do we think it's an interesting path to pursue?
So I guess that from my perspective, the fundamental question that's going to be asked in that regard is, does it give you a level of efficacy that you're not able to achieve with the microbubble? And there's some indication that that might be the case. So that would make it at some level application specific. But the other aspect of this is whether or not it's going to be viewed as any different from a regulatory perspective than coming in with a new agent. And that's going to be a question that's going to have to be addressed at some point in time. Because certainly using the diagnostic agents that are commercially available now and FDA approved for that purpose is a useful pathway. If the condensation of such agents and their application triggers a full regulatory path, then at some level, I'll just say you might as well be making whatever the new agent needs to be, right? So I think that there's going to be a lot of discussion along those lines. One comment that came through is the approved agents still have one big problem of polydispersion. Exploring further is always a good option. I would tend to agree with that, especially depending on the application. That having a more monodispersed population, there's certainly been some evidence in the literature to indicate that there are competitive advantages for doing that in terms of being able to limit, for instance, perfluorocarbon load and optimizing dose and several things like that. In addition to some of their imaging characteristics being advantageous in a more monodispersed population. I certainly would not like, I certainly would very much encourage the innovative nature of pursuing additional agents. I don't want the field to sit on the laurels, so to speak, of the agents that just happen to be commercially available. I think that would be a mistake for the field in general in terms of the development of agents.
Brian, the issue of polydispersity also very much impacts the earlier conversation about thresholds. That if we actually want to have clean thresholds on a per application basis, having an agent of the size and monodispersity required for that particular application is crucial. Part of the reason why thresholds are difficult to detect in microbubble formulations because of the huge range of agents and sizes that are actually involved in that formulation. So if an application is needed where there is a very sharp inertial cavitation threshold, you need a very monodispersed bubble population to achieve that. If an application is needed where you need a very sharp non-inertial cavitation threshold, having a particular type of bubble formulation or droplet will help with that too. And so as we start developing these monitoring methods as a way of monitoring treatment, having monodispersed tailored contrast agent or cavitation nuclei will help make those monitoring methods more relevant and more sensitive to detecting the onset of the bioeffect.
Yeah. Kostas?
Yeah, I also like to mention, like in addition to being able to select different agents for different bioeffects, I think when we use agents, is also important to report the dose, to report the administration protocol. There is a very strong discussion for bolus administration versus infusion administration and how this can impact both the emissions as well as the effect and as well as the kinetics of the agent. So I think that as soon as you go down a path with a certain agent I presume for very good reasons, I think it's also good to provide this information So people across different laboratories are able to better understand and also being able potentially to replicate some data. And also all this will affect the acoustic emissions for sure.
Yeah. And this could also help us to refine, going back to the previous comment, what should be the ideal characteristics for a given applications or given bioeffects based on the information that will be provided on the agents and the characteristics. Christy?
I want to push back on a comment that Constantine made about monodisperse nucleation agents. Though, you know, it gives you a very predictable threshold if you know that specific size. The minute that gas is liberated, everything changes. You know, they can grow by rectified diffusion, they change sizes immediately. And I'm not sure that we really need a call out for monodisperse nucleation agents. In my mind, having developed an MI particularly, I see an advantage of having an array of sizes so that no matter what you hit it with, you're going to get that bubble activity that you want. So I'm not convinced that we need to, other than you can size isolate or do this for scientific basic science purposes, but for the practical application, I personally don't think it's going to be a monodispersin size agent. Just an opinion there.
Thank you, Christy. So we have just a few minutes. Oh, James, okay, go ahead. And we'll move to the matrix.
I think Christy is right for particular applications, but we really don't know what the mechanism of action for many of our therapeutic techniques are. For blood-brain barrier opening, for example, it may not be just inertial cavitation, but inertial cavitation of a particular region. And the amount of mechanical stress that a bubble causes on that region might depend on its size. So initial size, for example, if you have a big bubble 4 microns in diameter in a capillary versus a bubble in a large vessel, you would expect different bioeffects. And then if you take a 1 micron bubble in a capillary versus a bubble in a large vessel, you may not expect the same thing either. And so although we focus a lot on inertial cavitation and whether that's happening or not, and that kind of stuff, it also depends on what we're actually applying the mechanical stress on, if that is what's causing blood-brain barrier opening, for example. And so I think size, we don't know, I would say. I think the answer is we don't know. And it could play a really, really important role, or maybe not.
Yeah, thanks a lot. And staying on the question of size, Kevin made a comment in the chat, and I think that's maybe could go into the challenges is how to accurately, let's say, measure the size distribution of the agents. And I think that's a very important point to raise, and at least to document in the studies in terms of what equipment has been used. I mean, what filter, for example, or tube, if you're using a Coulter counter, or what are the data, if it's average size in volume, average size in number, and things like that. So that's a very important point. In terms of, so going back to the matrix for the last minute of this discussion, if there are any challenges, I think, that we didn't address our list so far? Yes, Kevin?
I guess maybe building off that last comment too, I mean, I think a challenge is, do we even know what ways we need to characterize these? Like, do we want to say, do people need to report size? I mean, you know, as Christy was pointing out, perhaps many cases size doesn't matter. And right, there's lots of other properties of bubbles that could be potentially reported. Perhaps this is very application specific, I don't know. But so I see that as a challenge, you know, how do we report characterization of bubbles? Because there's so many things to potentially characterize about them. And not just bubbles, right? Contrast agents.
Yes, Constantine.
Kevin, I believe for the reasons James stated, size always matters, because in the same way as you can have inertial cavitation with an initial bubble size of 100 nanometers getting to 2 microns. You can have inertial cavitation with an initial bubble size of 1 micron getting to 20 microns. And I think if we really want to understand the whole purpose underpinning this session in general is for us to give ourselves as a community the tools to unpick some of the things we're not yet consistently understanding. So I think really having a good grasp on what type of cavitation behavior we are getting and where it is occurring is important. But for the same reason that you stated actually in the chat, I actually think having the size and size distribution and at least knowing what that initial size is important for 2 reasons. One, once we know the exposure conditions, it will enable the reader to make a sensible judgment as to whether nucleating a particular type of cavitation makes sense. So it will confirm in some sense the cavitation detection measurements. And 2, because the absolute size, at least the initial known size of the cavitation agent relative to the biological structure, probably has quite a lot to do with the resulting bioeffect. So I agree with your suggestion in the chat that we should agree, in an ideal world, a list of things: mean size, standard deviation, material properties, gas content, that always need to be included when referencing particularly a homemade cavitation agent. And when it's not a homemade cavitation agent, perhaps some specificity on the conditions on when the vial was opened and what the experimental conditions that might have affected the initial bubble population were. We ought to make recommendations in respect of all of those things.
Yeah, thank you, Constantine. I think that will kind of close the discussion for today about the need to provide some recommendations on at least how to report and to characterize. And so, Brian, we'll move to the last session.
Yeah, so in our final session, we summarized some of the clinical experiences that we have, and we started looking toward regulatory considerations. Obviously, we've hit a few of those regulatory considerations already, but we want to circle back to a few questions that the moderators posed regarding that particular session. So the questions are on the screen now. Where do the commonalities across modalities, the 10,000 foot view, exist? And I think that's a critical one, although it says it may be a possibly impossible question to ask. Where do the differences lie, and what will be carved out to be an application specific characteristic? And then how do we standardize calibration and reporting? And that's something that we heard a lot about this morning. So comments with regard to the questions that are posed here. I'd really like to see where people think the commonalities lie across the different applications. Well, I think that if we think about it in terms of device development, and if you're going to have cavitation monitoring as a part of that device development, I think that clearly becomes a commonality. I mean, the question of whether or not a given device is relying on bubble activity to be an end effector for its biological effect, that requires then that you understand the relationship between the cavitation measurements that you're making and the biological effect that's intended. So the commonality then becomes, can you at least be able to monitor the type cavitation activity that you have in a consistent fashion across the different modalities if they rely on that bubble activity in order to be their therapeutic device in order to be a therapeutic device. Suba?
Thanks, Brian. Yeah, I would just like to add to that, right? The commonalities, I think, also can include these questions, right? So is the device capable of generating cavitation? If it is, what is going to be used to detect it and monitor during treatment? So I feel like that is something probably any therapeutic device manufacturer would have to understand for themselves, and as well as understand in a regulatory aspect. So it's not, specifics, but at least starting with that question, with these questions, just like you said, in general, does it generate cavitation?
Well, I think it's a service probably to the field for those people that are developing the devices to be able to lay out what we think are the things that are going to be requested of them, right? Obviously,
right.
They can arrange a discussion with the FDA at any given point in time, and the FDA, I know from my experience at least, has been very open to the device manufacturers in terms of having discussions with them about what the expectations are. Providing that information so that device manufacturers know what it is that's expected of them is probably a useful exercise and a useful piece of information that we could provide as a part of the outcome of the workshop, for example, at least if not directly, by reference.
Agreed, especially because most of this is done on a case-by-case basis, and on our end, we would also like standardization to help our review, as well as help the sponsors know what they need to bring in to us.
Yeah, right. In terms of commonality, so maybe not on the bioeffects, but more on the needs for characterization, or maybe one commonality is that the need for very special information or special characterization of cavitation activity in order to differentiate the intended therapeutic use and the unwanted side effects, or in terms of cavitation out of the treatment zone. Or I think that's maybe related to the need for some sort of mapping, maybe not imaging, maybe not completely mapping, but at least some sort of triangulation or spatial localization in all the application that will use therapeutic cavitation.
I think that's a very valid point. So there's two sides to that. One is cavitation mapping as a means to be able to assist in the actual performance of the therapeutic treatment. And then there's the question of whether or not that type of localization is part of what might be expected from a regulatory sense. And especially when you think about the off-target potential, and whether or not can you guarantee that if you're using cavitation, is the cavitation only at the intended target, or is there cavitation that's occurring off target and you're not aware of it? And so the question then becomes, what is it that the regulatory folks are actually going to require in terms of understanding that? Because in general, most of the feedback in a lot of the systems right now are a limited number of elements that are telling us that there might be cavitation activity that's occurring. And it hasn't been that there's been a high level of specificity as to that particular approach.
Brian, just to add to what you said, there's also another wonderful thing about passive mapping cavitation imaging. is that actually it is potentially sensitive enough if the signal processing is done right to also detect that change in the original bubble size that Christy and Kevin and others have alluded to. So it may also end up playing a role if we understand it and we analyze it well enough at telling us when something has shifted in the injectable cavitation nucleation agents. And so the more robust we can make these tools, the bigger a role in treatment monitoring, but also in RA QA of treatments they might actually end up playing. And I think we will learn a lot from them if we can set them up correctly.
I think that's a very valid point. And one of the things that in the original figure that Fred put out at the beginning of the meeting, you notice that we progressed from mapping and detecting cavitation activity toward understanding the bioeffect that's being induced. And then beyond the bioeffect is the therapeutic endpoint, and that's that treatment feedback that you're talking about. And then in the end is whether or not you are giving any information that's related to the outcome of the patient. Because you're reaching a certain therapeutic endpoint and you're saying that, yes, I treated this. And then there's the real outcome. In other words, yeah, you treated, but were the margins treated? Were, all of those different components that are associated with therapeutic ultrasound or therapies like, radiation therapy all figure into this problem as well.
If I try to kick off maybe some discussion on the second point, I would venture a view which I know will not be popular, which is I actually think the differences between application areas are quite small. I think the difference is in what we call the good, the bad, and the ugly in each application, but the good for one can be the bad for the other and vice versa. So, we heard today about inertial cavitation in the brain being seen as bad news, but there are some drug delivery or surgical applications where inertial cavitation is a desired effect. I don't think of the methodologies we have been discussing, any of them are particularly limited or non-applicable to specific applications. But I would love to hear people's thoughts as to what they think is very special about subset of applications that needs separate treatment to all the others. Because I think that's something we need to try and capture.
Yeah, I would agree with that, Constantine. It's a very good point. And I would open it up a little bit further than that and say, tell me an application that's using bubbles as an end effector that does not need this type of monitoring, right? I mean, that would be the underlying question. Because in the end, if we say, well, every type of bubble activity based therapy needs some level of this type of monitoring, then yes, by definition, there is a commonality here and a need in the field. Now, again, it could be that 99% are that way. It'd be interesting to hear from people if they think there's any application where having this type of information is not going to be useful. And useful is a relative term, right? Adam? Oh, you're muted, Adam.
Sorry. Thanks. I think one that's I don't think it goes quite to the level of saying that this is not it wouldn't be useful, but one where I think it's been minimally applied is for histotrypsy and the various forms of histotrypsy, where a lot of the feedback on cavitation is from imaging, and it's not even necessarily imaging cavitation activity, it's imaging the location and the presence of nuclei that are that are in the field between pulses rather than the activity of the bubbles during pulses. Also, and so just take that to like an extreme case. In boiling histotrypsy, a boiling bubble massively grows and it never really collapses after that. And so the bubble is nearly static in that sense from like an inertial cavitation activity standpoint. But there's other activity that's happening that's producing tissue ablation that leads to histotrypsy. And I think the same might also be true in therapies as well. And the other extreme is intrinsic threshold histotrypsy, where the pulses are so short and the activity is all occurring in like a temporal on a temporal basis rather than looking at something like a frequency domain analysis of the cavitation activity. So you're looking at specific physical phenomena like bubble collapse, which has been one proposed metric of cavitation activity for that particular modality. So just, I guess, to summarize these kind of extreme cases, they don't rely necessarily on quantification of inertial cavitation activity so much. And again, it's not to say that it's not an important factor, that it doesn't have some benefit, but historically, the spatial information and the presence of bubbles has been the driving factors to whether histotrypsy is going to be effective in a certain case.
That's an interesting point, because it points then to whether or not passive cavitation monitoring it's a tool, but it's not the only tool that might be needed in therapeutic ultrasound going forward, right?
Yeah, I think another subset of tools, in addition to the quantification of the actual dynamics of the bubbles, is related to the spatial position of the nuclei, or the spatial position of the bubbles, the density of clouds or groups of bubbles as well. How well we can detect all those is another question, but there's things like active cavitation detection using Doppler, where they're not actively sensing the bubble activity, they're just sensing the presence and location of nuclei that could potentially produce cavitation.
Yeah, that's a good point. So there may be opportunities here, and Fred, you can flip to the next matrix slide for considering things for the future that might help characterize the bubble population. And I would add to that, possibly measure characterizing the bubble population in situ, even for exogenous administered contrast agents, if there were means of being able to adapt ultrasound fields to take advantage of particular populations of bubbles. You'd like to know kind of what that population is, and then to Christy's point earlier about, well, once you liberate the bubbles, then now you've got a whole new population that exists that are different than what you were looking at previously. And so any subsequent application of ultrasound while those bubbles are still present in the field then becomes something where maybe that type of feedback could be useful, so that you have some idea of what that bubble population is. It may be a little bit futuristic, but it might be useful to the field in terms of opportunities that will exist for being able to pursue this. So any items under this category that we've not captured, any final comments with regard to either knowledge gaps or opportunities or challenges that might exist in this space? Okay, so this has been a good set of robust discussions about each of these topical areas. Now we're going to move on to part two, which is some discussions of the potential partnerships that we might have with different organizations. We've got a couple of representatives here that we've invited to participate in the discussions. And so, Fred, I'll pitch it back to you for beginning of part 2.
Thanks, Brian. So we invited, as you said, different people to share some experience and information. So maybe so the idea, is to maybe to start the conversation about how should we move forward to elaborate, to create, to write some standards on cavitation detection. Maybe will be the first step. And this is for that we invited people external to the to this specific workshop to share expertise. And maybe we'll start with Suba. You wanted to share a few slides about the IEC committee to explain to us how it works. And
can you see the slides?
Yes.
Yes.
Okay.
Yes, we can.
So yes, I'd love to just give a very brief overview of IEC TC87. It deals with ultrasonics. So the scope is this: to prepare standards related to the characteristics, methods of measurement, safety, specification of fields, equipment, and systems in the domain of ultrasonics. It's very wide-reaching. Excluded from this scope are safety standards. However, the safety standards usually will point to one of the standards developed within TC87 for, again, exactly how to make the measurements or other such issues regarding ultrasonic equipment. The safety standard.
Suba, would you like
Yes.
to put your slides in presentation mode? It'll make
Sure, I can. Yeah, no. How's that?
Thank you so much.
So the safety standards are maintained by a different group, TC62. We have, quite a few for different medical ultrasound equipment, such as diagnostic, physiotherapy, HITE2, and lithotripsy. The structure of TC87 is quite broad. We have volunteers from about 14 participating countries. We also have observers from 18 different countries. And the structure of IEC documents is important as we move forward. There are 2 possible paths. One is the international standard. This is a normative document developed according to consensus procedures, which has been approved by IEC National Committee members. And the second is a technical specification. It's published when the subject under question is still under development or when there is insufficient consensus for approval of an international standard. And sometimes technical specifications will become international standards when there is consensus. There's also the technical report. This contains collected data of a kind different from that normally published as an international standard. Data obtained from a survey carried out among national committees, data of work in other international organizations, data on the state of the art in relation to standards of national committees on a particular subject. And it's entirely informative, no normative material. And then there's the new work item proposal. So any document starts out as a new work item proposal and then can go either the technical report, technical standard, or international standard. This is an overview of the TC87 working groups, and I'm so glad that many of these conveners are actually on this call. Working Group 3, high power transducers, is convened by Sam Howard. Working Group 6 is HITE2 and focusing transducers. Tom Szabo is a convener. Working Group 7, surgical and therapeutic devices, Friedrich Oberle. Working Group 8, ultrasonic field measurements, is Volker Wilkins, who's also TC87 chair. Working Group 9, pulse echo diagnostic equipment, Zhengfeng Liu and Peter Edmonds are the co-conveners. Then there's Working Group 14, of which I am the convener, determination of ultrasound exposure parameters. And Working Group 15 is underwater acoustics. Stephen Robinson is the convener. And this is another group that might have some expertise that could be useful for cavitation detection. I'd like to say also that Peter Edmonds did mention that there might be potential IEC standards that come out of this workshop for terminology, transducer and field calibration, microbubble detection, microbubble field mapping, quantitative analysis, thermal ablation, specificity towards histotrypsy and lithotripsy. So while I propose just a very basic cavitation detection standard, there are quite a few that we could work on in parallel. So that's kind of all I wanted to share. I didn't want to get into the actual documents under each working group, but if anybody would like more information, just please let me know. Happy to share.
And Suba, just to understand to work toward a standard, you need the technical specification document. Is it the first step before you move toward the standard, or?
Right. So the first step is making sure that there is enough material that has been published or disseminated before we actually even put that standard together. So there definitely is, I think, with cavitation detection equipment. The next step is forming a working group. Now, with cavitation, I think, extends across all of these different working groups. So, we've been talking amongst, IEC conveners and the chair, and we think that it is important that there is representation from all these different working groups, that it doesn't just come under one, especially as we're putting this together. And we may decide that, we are working on a couple of different standards in parallel and, can redistribute as needed. I feel like a great starting place is this workshop. I think we've got our conveners, working group 3, Sam Howard. He's in charge of a standard for cavitation measurements for ultrasonic cleaners. That could be a great place to start. Working group 6, Tom Zabo, I know that they're already dealing with cavitation, and it's certainly mentioned in the HI2 safety standard. Working group 9 has a vested interest in pursuing cavitation in a new standard, as does working group 7, surgical equipment. Lithotripsy comes under working group 7. Friedrich Uberle is here as well. But I think, before it's even submitted to the IEC as a new work item, it's really helpful if we have a near finished draft. And so that can be done outside of our regular meetings. IEC usually meets TC87 usually meets every 9 months. And every year and a half, we have like the formal meeting. We have interim meetings every 9 months. The formal meeting is every year and a half. But I feel like one thing that we could do is start, Zoom meetings with those who are interested from, the external community, as well as IEC conveners and people within our working groups who are interested. Begin discussions on just organization, content, references. I think that's going to be very important, that we identify all the references needed for the standards. I invite Peter Edmonds, Friedrich Uberle, Sam Howard to comment as well if I've missed anything, but it is very helpful to get, like a near done draft. Then it is submitted as a new work item. And then after several rounds, 2 or 3 rounds of comments, like as a CD, CD2, and then a CDB where it's near completion, it then will get published as one of these, either international standard, technical specification, or technical report. And it is worth considering that that we do consider one of these 3 paths, not necessarily an international standard. A technical specification, technical reports could take, they might take a less time, sorry, than an international standard.
Okay, thanks a lot.
If that is of concern.
Brian, do you want to introduce Peter?
So I know Suba referred to Peter Edmonds, and also Sam Howard has been on the call. I don't know if he's here right now, but obviously they've been involved at the IEC as well. They may have a couple of comments and that might be of particular guidance for us. Peter?
Thank you, Brian. I would like to remark that it is necessary to establish agreed terminology in order to proceed with much of the following technical development of a standard. And this is a difficult subject because everybody has an opinion on terminology. And so a terminology working group attracts large numbers of participants, and its activities can last for decades. In the case of the IEV document, which is now on the IEC website. IEV stands for the International Electro-Technical Vocabulary, and it's a special publicly open website where anyone can refer to the terminology that has been used for any of the published IEC standards. IEC standards otherwise are restricted to those who paid to buy them from the IEC. But to come back to the question of terminology, the example I was going to cite is that of the terminology document for ultrasonics, which is number 802. And the working group for that existed for 22 years before I drove it to a conclusion and we got a document. I hope you won't repeat that experience.
Thanks, Peter. I completely agree. And we alluded a little bit to that before, but I think that IEC has kind of renewed their commitment to be a bit more nimble than that experience. And Ron Roy actually pointed to an ANSI experience that we had in the cavitation field several years ago, about 2002, where the birthing of a ANSI report took several years, not 22, but it did take several years to get to that point. So I think as much as we can leverage information that already exists with regard to terminology and try to reach consensus on that, I think we would further the field much more rapidly. So that's a critical point in that regard. So Suba, I saw you.
Brian, yes, can I add that the purpose of getting a near draft done before we even submit it as a new work item is that it will go through the different formal stages of IEC approval much faster.
Yeah, and that's been
kind
of yeah, exactly. And that's been kind of, I mentioned in the email at one point that for example, the AIUM Technical Standards Committee and other organizations like that have kind of taken on the role of trying to get things to a well-developed point to be able to send them forward at the IEC, and that kind of taking the commitment of being able to develop international standards so that people don't have to deal with a standard in every country type approach has been something that we've tried to drive as well. So I think this group or some subset of it would be very, very useful for trying to identify what I would call the low-hanging fruit, the things that we can get ready and get before the IEC in some reasonable time frame. Sam, I see that your hand is up.
Yes, thank you, Brian.
I just want to reiterate what Suba and Peter said and just touch upon some of the distinctions that can help facilitate the passage of a standard. A technical specification requires fewer yes votes, and so it's sometimes faster to get that approved. The trade-off is there's an expectation that it's going to be updated sooner than a standard, but they both have potentially equal weight. And the other thing to bear in mind that I want to mention is that I believe there are other options for doing standards, the Acoustical Society, et cetera. But what seems to carry most weight in industry for various commercial and regulatory reasons is the IEC standards. Typically, those are what various notified bodies look for when reviewing industry applications and things like that. So those are just some points that I just wanted to add.
Thanks, Sam. And that speaks to the point of trying to get to the international point. And obviously the collection of people that we have involved with this type of bubble activity monitoring and therapeutic ultrasound applications is clearly international. I want to introduce Stephen Rusek, who is with the National Institute for Standards and Technology. He's joining us today because of the interest that NIST might have in being able to be involved as one of the potential partners for an activity like this. He currently leads the imaging physics project in MRI at NIST, but has been involved in discussions with KIBA with regard to different aspects of being able to provide resources associated with standardization activities. So Stephen, you had some thoughts to share with us today?
Yeah, let me share my screen first of all and see if I can. I'm an MRI guy, and so I appreciate the opportunity to sit in on your workshop. There is some overlap in some areas of partnership. So can you see my screen?
Yes.
So I work in the medical imaging metrology and standards area, and we have two things that, two services that ride on top of that. One is a biomarker calibration service where we provide traceable values for things that can be measured by medical imaging. And so these are measurements where, you know, we understand all biases. The actual bias is less than the variance. So these are ground truth measurements. And I'll show you an example of that. We also have another service that's a phantom lending library, which is a great way to validate and curate standards for long periods of time. And this work is also sponsored by NIBIB, and we have lots of collaborations with NCI and RSNA. So NIST has been in the business for quite a while. So here on the left, you can see some calibrations of medical thermometers back in 1905. And back in 1905, just getting people to agree on a common temperature scale was a big issue. And internal measurements of temperatures are still a big issue 120 years later. So the middle one shows some work on calorimetry for CT dosimetry, which is a better way to go than air kerma. So this is a very challenging area. So for radiation therapy, you typically will get tens or 100 millikelvin temperature rise. For CT, where the X-rays are only 120 keV, the temperature rise is in the microkelvin range. So it's a really tough metrology to do primary calorimetry for CT. And this has been a very long standing effort. My work is in MRI standards, and that's really pretty new. We started only about 15 years ago. And again, what we do is we make primary standards and calibration objects. And what we do is provide traceable ground truth values for what goes into the phantoms. And that's sort of a unique role for us. So the Phantom Lending Library, you can go online, you can click on it. You can actually see what phantoms are available. There's a calendar. There's a communal database where you can see everybody else's images on the same object. There's also communal analysis code. It's fairly easy to use. Typically, if the phantom is available, we ship it out overnight, and loans are from typically 2 to 6 weeks. So this is sort of our roadmap for our phantom lending library. So these are the existing phantoms, which are pretty much all MRI. We have a breast phantom, a system phantom, cardiac and diffusion phantoms. The yellow stars are the ones that are developed in collaboration with the National Cancer Institute. The red stars are the ones that we're doing in collaboration with RSNA KIBA. So they're all very communally developed reference standards. What we do is we try to maintain, validate, and curate them so that they don't come and go, and somebody's actually making sure that they have the correct properties. So what you can see there's a migration here to more sophisticated phantoms that are more biomimetic. And so what we're trying to do is incorporate structures that look more like neural bundles, more like tissue, more biomimetic. And that means that the phantoms are getting inherently multimodal. So if you make a really biomimetic phantom, it doesn't matter whether you image it with CT or ultrasound or MRI. And this is one that we're looking at. It's a triple modality abdominal phantom, which you can image with ultrasound, MRI, and CT. And you can also use it for therapeutic education. You can use it to test image-guided biopsies. And the key is to get these phantoms so that they have tissue mimicking properties. They're stable, they're reproducible. So we're sort of moving in this direction. We're developing biomimetic tumor mimics, again, that have that are both stable and have they basically mimic multiple tissue parameters. And then in the future, we're going into basically mimicking cellular structure for micro MRI and micro CT. So when you get over here, there's going to be a lot of overlap with both CT imaging and potential therapy. So now I'm having a hard time advancing it. So I just wanted to show you a couple of uses for our phantoms. This is a multi-site round robin that's comparing a measurement of T1 over multiple sites and these are averaged over the vendors. So these are the values for the different vendors. The gray box is the NIST value. So this is the ground truth. This is our fundamental uncertainty. So here you can see that for MRI vendors, there's systematic deviations in scanner measurements that are beyond the fundamental uncertainty in the properties of the phantom. This is another example which shows the development of an advanced MRI technique. This is called MRI fingerprinting. It's a much faster way to measure multiple parameters simultaneously. And the key is you need to be able to develop it and validate it very quickly. And you can use these traceable phantoms to do that. And this shows you some measurements, long-term T1 and T2 measurements that on our traceable phantom with this new MR fingerprinting technique. So the phantoms are getting to be pretty complex. This is a breast phantom that has a lot of tissue and fat mimics. And this really comes to the crux of standards, is how do we standardize materials that are tissue-like, very complex, have multiple parameters. From an MR point of view, we have lots of different protons in there. We have lots of different relaxation times. And so the question is, if we really want to mimic tissue, which is eventually what we want to do, it's very hard to provide precise reference values, and that's something that we're working on. I really think that multimodal imaging and therapy is the frontier, which is why I was very pleased for letting me sit in on your workshop. So on the MRI side, there's MRI guided high intensity focused ultrasound, and the measurement parameters right now are typically temperature using proton resonance, frequency shifts. There's many other potential thermometers, but there's a big need to validate and make these temperature measurements more precise. But also there are a lot of other biomarkers of tissue change that you can use to see what's going on to the tissue using the high intensity focused ultrasound. And there's a lot of additional information that we can get not just temperature, but what happens, after exposure. What does the tissue look like? There's diffusion and many other parameters that can help understand what's going on in the tissue. There's magnetic resonance elastography, which is a low frequency, sonic excitation to measure shear waves that propagate through different tissues. That's coming along. It's going into sort of vector elastography and moving to more challenging organs such as the head, and you get out, stiffness maps. You can get out lots of more properties. But again, this combination of modalities is really where you get a lot more power. And then there's MR detected microbubbles. We did some experiments back in the day where you load the bubbles with MR contrast. So not only can you see the bubbles, but you can actually see how they deliver payload. So you can see the gadolinium potentially as it goes through the blood-brain barrier to see how your therapeutic is being delivered, and you're again imaging in MRI. So anyway, so summary, you can pretty much read it. We have some resources, Phantom Lending Library. It's MRI centric right now, but we're going to expand to CT. Ultrasound is also an area that we'd like to expand into. There's a lot of advantages to having these type of systems. You have, you curate standards for long periods of time, you validate them, you provide SI traceability, which means that you get rid of all biases. Whoops. So this is sort of the NIST niche, is to help people out maintaining standards, providing traceability. So that's about it. So I appreciate your interest and potential partnerships.
Thanks, Stephen. Any questions for either Stephen or Suba before we take a short break? And while we're doing that, if you could release the screen, Stephen, and then we'll, and Fred, you can bring your slides back up. There's a partnership slide that Fred's going to show. Questions from anyone? So the reason that NIST got involved with the Kiva activities is because of the potential of being able to create these reference standards in the reference library, Phantom library, that might be beneficial, again, as we perhaps it's part of the activity, then settle on particular types of instrumentation and calibration for that instrumentation and what's going to be required in order to do that. This is a slide that we put together just outlining a few of the partnerships that we know might be of interest to this activity. We know that there are a lot of different stakeholders. This is not a complete and exhaustive list. And we plan on sharing this with our organizing committee and getting input for other partnerships that we might want to consider. As been pointed out at various points during the workshop, it's going to require additional expertise to be able to pull in people with biologic knowledge and different types of knowledge that we're going to need, as well as resources for being able to conduct any things like round robin trials that we might want to consider as a part of the activities. So on our schedule right now, we're scheduled for a short break. Maybe we'll make it just a 5-minute break for everyone. And then the important third part that we really want some input on in these activities is kind of a series of next steps. And we've got an outline to kind of follow to guide us through that discussion. But we really would like people's feedback on what they think is important to do, both in the short term and in the longer term, so that we can begin to prioritize those activities. Okay, so we'll take 5 minutes. We'll see everybody back then at 12:25 for the final portion. Thanks.
Okay, I think we are ready to start the next and the last part of our discussions. Can you see the screen?
Yes.
Yeah, okay, super, thanks. So yeah, this is a slide that we put together to guide the discussion and the different items that we wanted to discuss. So in terms of next actions, the follow-up discussion, what will be the output of the workshop, our actions toward standard development, and discussion about resources that will be needed to successfully respond to all these items. So the first point of discussion is how are we going to continue the work and the discussions in terms of, so do we need, do you think we need to set up specific targeted workshops, for example, on very, very specific topics? Do we need to organize roundtables, for example? That would be also another way to continue and foster the discussions. Do we need to set up working groups on specific topics? So this is again, this is open to discussion. Obviously, there will be some efforts for standard development, so that we need to create this working group. This is one aspect, and we can review it later. But in terms of continuing the discussion about the scientific evidence and the work that is needed. So do we need, do you think, additional working groups? What's your, we'd love to hear your ideas and suggestions.
So Fred, I'll kick it off.
Sure.
So it sounds like from talking about especially the calibration and detection and those types of issues, I think being able to leverage additional expertise that's out in the field that has been doing calibration problems, for instance, in diagnostic ultrasound and different areas like that, along with the IEC standards and other types of information that's sitting out in the field, figuring out what is it that is particular to the therapeutic ultrasound problem that is unique for being able to make the right measurements and the right standards. I don't want us to reinvent the wheel, and yet I know that there is a lot of work that's already been done specific to therapeutic ultrasound and the problems that we talked about in the first and second workshops. But at the same time, I also know that there's a whole bunch of stuff that's sitting out there, and I don't know where those two overlap. So I really think that there's a need to do some careful study about what's already happening in the standards area with regard to diagnostic ultrasound and even therapeutic ultrasound, and see how those things then relate to the specific problem of monitoring bubble activity. Because I think a lot of the things, especially in terms of calibrating transducers and those kinds of things, there are unique aspects, and yet there are aspects that have real commonality for other modes. And I'd be interested to hear people's thoughts on that. I know Peter Edmonds, in the chat, you triggered a link out there to some of the IEC stuff that might be relevant. So I'd be interested in people's comments that are familiar on one side or the other with this particular question.
Yes, Sam, go ahead, please.
Yeah, so I agree with you, Brian. there are a lot of things out there, and then at the same time, a lot of us who are familiar with the standard approaches for transducer calibration are less familiar with these particular needs. So I think having a group where you have both sides come together will be useful, where some of us who are familiar with methods of calibration that are more standard right now can talk about what's available and understand better what the needs are and perhaps tailor some recommendations. Keeping in mind, of course, that in parallel, there are some new things that can be developed, yet at the same time, there may be some interim old approaches that can help.
Yeah, that's a good, thanks, Sam.
So I think establishing maybe a working group that's associated with that could guide us on whether or not, for example, are we in a position to be able to reach consensus on what types of measurements are needed, as well as what's already possible. And frankly, I don't know, and there are experts on the call that are much more familiar with this, how unique this particular problem is. I think that it's a problem of specificity to therapeutic ultrasound, and more specifically monitoring bubble activity, versus general calibration of the sensitivity of array transducers, which is not new. But at the same time, I don't know what necessarily are the big problems that exist in that calibration that might not be otherwise addressed in the methodologies that are out there now. For instance, I know bandwidth is an issue. Constantine?
You're right to state that I think from a metrology perspective, cavitation is quite an unusual problem, because I think it's quite unusual by the number of its shades of gray. And what I mean by that is you can have, there's no such thing as a standard cavitation source. What you create and what you observe depends already on three things: the incident field, the cavitation nuclei that exist, and then the receiver. You can have different frequency domain responses. You can have different behaviors, even within the same experimental setup. I think what I'd love to hear from the people, the speakers of this last session, is whether there are other contexts of such multifaceted standards problems that we could draw inspiration from in terms of how to deal with the multidimensionality of the problem. Because I think actually ultrasound, pun intended, has certain shades of gray. So calibration standards in ultrasound are highly relevant, but in my view, not as many as this problem we're working on. And so are other fields in MRI, in X-rays, where this multidimensionality has been encountered, and what have other communities done to address it? And I'm particularly referring to standards here, right? So how do we get to standards for something that has so many degrees of freedom?
Go ahead, Suva, please.
So Constantine, I think one place to start is looking at how the hydrophone standards are developed, right? There are quite a few. They come under working group 8. And you have them covering the different bandwidths, as well as how to report. I'm just looking for 62127. There's 62127-1-2-3.
Yep, very familiar with those.
Right? Yes, right. Okay, good. So that could be a place to, just to see how, how they have kind of organized that material. I think a lot of this is about organization, right? content and organization, like where do we start? And I think that is a good place to start, as well as the 63001, Sam Howard's standard on cavitation measurements for ultrasonic cleaners, especially in terms of terminology, as Peter Edmonds pointed out, because that's another thing that we kind of have to get consensus on early, is what are we going to be defining and how do we define them, and then get into, you know, the calibration, the characterization, you know, those issues, right? So that could be a good place to start. I don't think it'll take us 22 years, but I think we can cut that down. I really do think that we need to leverage what is out there. I mean, that's how standards are formed. They are formed from, you know, the material, the published material, right? So maybe another place to start is combining references that have already described how to characterize cavitation detection, and then pulling that with the terminology that is already in place and what still needs to be, what we still need to get consensus on, because there are definitely terms that people define in different ways. So we need to identify that, you know, those terms. And so that could be a place.
Super, that's a great suggestion. Thank you.
Yeah, so it sounds like then maybe one of the working groups that really needs to happen is this one that would deal with the terminology.
Mm-hmm.
If that is kind of a bar that we have to achieve, that commonality in that and consensus on terms, in order to be able to go on to the writing of any subsequent standards or technical bulletins or technical specifications or whatever, then that's something that we probably should jumpstart and get started quickly. We can draw from the previous ANSI technical report that was done on cavitation monitoring or bubble monitoring. We can also, like you say, pull from Sam Howard's materials as well, and any other that are related to that type of bubble. And even gathering those bits of information could be critical to getting the kind of uniformity of what the language is going to be with anything that we would write subsequently. I see that Sam Howard is in agreement.
I am entirely. I also wanted to add, I think another experience from IEC is, insofar as possible, in terms of your organization of the work, take a divide and conquer approach. For example, it might be good to divide the question of calibration in situ or in tissue mimics from calibration in water, so that when you're talking about in water, you can focus on that and you don't drift off in terms of the discussions. There are choices to be made about that, because when you're doing that, obviously you may miss important issues, but it's important in organizing the work to think about that, because there are some things that there is existing literature to leverage off of and other things that aren't, and you want to organize the things where there isn't the existing literature in one space and then try to drive towards some conclusions on the more standardized things.
Right. Circling back to Constantine's question of the multivariable problem, I know, Stephen, in the MR space, even the fingerprint example that you gave was giving multidimensional data out of that. Do you have any comments on strategies that might be related to that?
Yeah. Well, first of all, yeah, I mean, I feel your pain, but you need, you start simple, you define parameters that you think you can measure, and then as you work into it, you find that you need to redefine and make things more precise. So a lot of the MRI side, they're measuring parameters, and at some point we say, well, that doesn't make sense. You just haven't got the right parameter set. You need an expanded parameter set, and then you have to redo that infrastructure. And so that's part of what we do, is we sort of figure out when, when you are at the limit of, your model, and you can no longer use, a simple set of 3 parameters to describe your tissue. It doesn't work. And so, you start simple, and then you just make it, you got to, incrementally make it, your model more complex and more precise, and understand exactly what parameters you're measuring. And I would suggest, certainly for bubbles, I mean, a multimodal approach. I mean, we characterize bubbles with MRI all the time, and micro CT, and at many different length scales, and we look at dynamics. And so, ultrasound, needs to be combined with, all the other modalities, and sort of think of, all of these things as combining, additional information to really try to, precisely define, what's changing, what's occurring in the tissue. But you do need to start with, some simple, well-defined parameters. And then as you work through it, you find that those aren't sufficient, and you need to get more complex.
So then start with a spherical cal.
Right.
Yeah.
So for instance, for us, a key MRI biomarker would be a relaxation time, T1. And so you say, okay, this tissue has a T1 and a T2. And you say, well, that doesn't really work. It has a spectrum of T1s and T2s. So you need to describe the spectrum of that. And then of course you have protons on, on water and on fat. So then you have to specify it versus frequency. And then you come up with a description of tissue, which, you can measure, and you end up doing something similar to fingerprinting. You say, okay, here's my fingerprint. Tell me what the tissue is like, as opposed to saying, this is the parameter that describes tissue. I have a complex fingerprint. And then you go, okay, now I know what the water density is, the cellular density, the integrity of the cell membranes. So it gets to be very complex, but that's sort of where the field is going. Instead of having 1 or 2 parameters, getting these very complex fingerprints, and then backing from that to the actual tissue parameters of interest.
So that might fall under the paradigm of spectral content, for example, for cavitation noise. So if there are signatures that exist within that spectral content, that might be indicative of particular types of bubble activity, or might correlate with a particular therapeutic endpoint or biological effect that is the desirable biological effect, then that's when that commonality of measurement matches up against trying to figure out what the biological impact is of that particular signature.
Yeah,
that makes some sense. That's an interesting perspective. I do want to be mindful of the time.
Yeah.
Are there other obvious in this category, say, additional working groups? I've identified possibly a terminology working group, and I saw, Peter, your call for somebody to revive working group 13. There might be somebody who's willing to convene that once we get to a point that it makes some sense to do so. I think we're in an information gathering stage, although, it takes some time to actually rev something like that back up again. And so maybe an emphasis in working group 13 targeted specifically on this calibration related to cavitation activity might be appropriate for upgrading the terminology. And certainly we would want to look at the terminology that exists at IEC already with regard to ultrasound, whether it's related to cavitation or bubble activity or not. Those are terms that we're going to have to agree that we have to use going forward, or adapt them in some way. And that would be where any changes that would be necessary at working group 13 in terminology would be necessary.
Yes, Kevin.
Oh, go ahead, Fred. Oh, Kevin.
Sure. Yeah. So I fully agree the need for consistent terminology, particularly as we move to standardizing things. At the same time, though, I don't think that we should wait on that to have things moving forward with ideas on how we practically carry out some of these calibrations that we've talked about.
Oh, I completely agree. If we don't do some of these things in parallel, then it becomes, we create a rate limiter. And that's not going to serve the field well at all. Suba?
I volunteer to just on the IEC end with the documents that are already that are already published, just kind of note where cavitation is mentioned and put together kind of a list. I did that for just the safety standard, but I know that it's mentioned in a wide range of standards across TC 87 cavitation. So I'll kind of start putting that list together. Just to understand where it's mentioned, how it's mentioned, and yeah.
Okay, that's awesome. Thank you.
Yeah, thanks. Yeah, so we probably have already a few working groups that are in the making. So to try to summarize, right, terminology, review of maybe existing standards in terms of what we need to improve or develop compared to what is already existing, discussion about parameters, what needs to be measured, and starting also drafting this our methods of calibration. So maybe one way to organize all of this would be we can through the foundation or IUM, we'll see collect. We have the email of all the participants of the workshop, so maybe we can create a kind of a newsletter or email list, and we call for action or volunteering for these different working groups, and start from there. And we can regularly update everyone on the progress of the future meetings or actions. So that could be one way we could structure this. Maybe we need to move to, what time is it yet? To the next item, which is the outputs of the workshop. So we intend to write a white paper. We will probably do that with the organizing committee, based on the summary, for example, that have been shared for each of the session, and the idea of the white paper. One of the idea of the structure of the white paper would be to have, for example, 2 pages per session, so for each topic. So again, to try to summarize very broad view of the discussions and the state of knowledge and gaps of knowledge, with a conclusion discussion sections to highlight the different needs maybe and future actions. So that's one action, I think, Brian, that we will for which we will take the lead, right, and try to organize it with the organizing committee. But we have a lot of material that have been accumulated during the workshop, all the presentations and the discussions. So the question that is open now is do we want, or do people want to use this, and based on this to start maybe drafting review papers on more specific topics, for example, equipments for session 1, cavitation mapping localization, for example. That could be maybe a first a very good way also to accumulate all the references that will be needed for a standard. So yeah, so open to suggestion and comments, and maybe that could be led by the moderators of each of the session. Yeah, open to discussion, and we'll be happy again to support any initiatives. Yes, Kevin?
So I like your idea quite a bit, Fred, in terms of, having kind of an overarching white paper and then potentially review articles, with potentially the moderators kind of helping guide each of those review articles. I do think in the context of doing that, obviously the speakers and the panelists are natural individuals to contribute to that. But if either the Focused Ultrasound Foundation or AIUM is going to kind of create an overall list and invite people to kind of participate in some of the other stuff, the working group stuff, I think we should put out a call for people that would also want to contribute to those review papers. Because while we were necessarily limited by the number of speakers and panelists we could have on each of those topics, and I know there are some great people out there that weren't included just because of those time limitations. So I'd like to see it, I guess, opened up more broadly.
Sure, of course.
I would completely agree with that, and Kevin, you're spot on. I think when we start talking about it topically and thinking about how to gather the knowledge that we have in the field, this workshop was supposed to spark that activity, right? And you're right, we were only able to have a certain amount of time devoted to that, and a review of a particular area, topical area, where the moderators for that can take a lead and solicit additional authors and contributors for that particular area is only going to strengthen kind of the final outcomes and knowledge that we have and help identify the gaps, right? So this first white paper is supposed to just kind of give the general things that were discovered or discussed in the workshop. But I think it's going to take a deeper dive, especially to reach the level of information that's going to be needed to drive any standards consensus.
Excuse Kevin, go on.
Sorry, I don't mean to dominate here, but is it, because I'm not very familiar with the standards process, is it helpful or harmful if, for instance, before a standard is published, if a guideline article, for lack of a better term, was published, right? So I'm kind of thinking there was the IEEE special issue a few years back, Alfred, you and John Hasek did on kind of methods, right? And so people published methods-oriented things. And so I could see like a methods-oriented guideline article for, any number of the topics that we've covered. It would perhaps help give some direction to, subsequent, more rigorous standards that would be published. At the same time too, though, I don't know, based on how standards, get developed, if that actually would be more harmful. So perhaps some of those involved in standards could comment on that.
It's helpful, Kevin. Again, because standards does rely on methods and data that has already been published. But I would say that I do think that there is enough that's been published regarding just basic cavitation detection equipment that we could just start compiling those publications and get started on a draft. I don't think that's the case where some of the other standards that we're considering are I think that some more work might be needed, but at least for a basic equipment standard.
Right, and I'll put my kind of academic hat on just for a moment and say that if you're going to engage people in this type of activity, I think being able to get a peer-reviewed publication, even as a review article, as a consequence of that activity, is highly valued. And while we all volunteer to provide help to develop international standards, the degree to which folks can get, how shall I say, academic credit for that development, that's facilitated and aided by peer-reviewed publications in that process. So being able to leverage those then peer-reviewed publications in any standards development, I think is going to be extremely useful. Keith?
Yeah, thanks, Brian. Yeah, I was the moderator on the 5th session, Clinical and Regulatory Considerations. I would be glad to lead the effort to write a review article on that aspect.
Okay. Well, we'll probably tap you first for kind of the 2-page that would go in the overall. And then, but we really appreciate the volunteering to do the deeper dive on a review article related to that. And I think these things are really going to help us move forward.
Okay, I think any more comments on this, on the output of the workshop? Oh, see, one other item I wanted to discuss is the what do you think we should do or we could do, or again, to exploit all this material that has been shared through the presentation? And one of the, I think it was Christy who suggested, like one of the session, we could use it to draft a course on, I don't know. So is it something that you think would be valuable to use this material to derive? Yeah, again, would be a course online with the, watch the video, answer the questions, something like that. Or another thing that the foundation, at least from the foundation side, typically does is that the website will be active for a few months, I think, and then very often we transfer the videos to our YouTube channel. So that's another way to share this material. But there are any other, so besides publications, let's say, ideas or suggestions on how to what to do with all these materials? Yes,
Frederic, having attended all of the sessions, I think there were some really great tutorial lectures given. I think people put a lot of effort into providing not just a normal research talk, but kind of a state of the art. And I can think of many starting graduate students or postdocs moving into the area who'd actually find them a very useful resource as a starting point to start their own literature reviews. So I think it would be great to create a repository that can be linked to from different places, organizing the videos by topic, perhaps with a simple summary of what each of them contains or focuses on, which could be written either by the authors or by the panel, by the session chair, or by the foundation. But just sort of keywords that would point people to which of the videos is relevant and I think the main thing is to make it searchable. So the problem with videos is they end up in a place where unless you know what's in the video, you can't actually get pointed to it. So if we can create suitable metadata for each video, I think they will be used, and I'd very much hope they will be used.
Yeah, that's a very good idea to make it searchable. Yeah. So, Christy, yeah.
So there's been this effort by Gail Tahar to put together a summer school on therapeutic ultrasound. And I actually think you could have a summer school, particularly for graduate students or postdoctoral fellows that are new to the field or newer to the field. And then flip the classroom for that summer school where the videos of this workshop essentially are your homework. And perhaps many of the people that gave those talks would be present for a Q&A, and that's where the real learning comes in, that interaction with the people who have pre-recorded this content. I'm thinking it would last at least a couple days, maybe more. And then, so therefore you have some online content as well as some in-person content. Now, this is all assuming that we can get beyond the pandemic, but I actually think this would be good to engage the next young brains on this topic.
It's a very good idea. Thanks. Yeah. Okay, maybe we need to move on. We have just 10 minutes, 15 minutes left. So the next item was standard development. I mean, I think we covered a lot of this already in terms of what will be the next step. So probably these working groups. Is there any other actions that we should take toward the standard developments? No, nothing to. Ryan, do you want to?
Well, I would say that I think we have spent a good amount of time thinking about this particular problem. I think since this is a very international group, there may be opportunities within a given region for thinking about, leveraging materials that might exist at different organizations that might be related to this. I know I mentioned the ANSI document. For all I know, there's an equivalent document that's sitting out there in the space related to the European Union or countries within the European Union, for example. Maybe similar things related to Asia or other continents. I think that making sure that we have the scope of things would probably be the most important. And that's the reason I put kind of the agency engagement there. I would add to that then, groups like NIST. Obviously NIST is the US version of this, but we know that there are national labs and things that exist at different places around the world that may be able to contribute similarly in terms of the development of standardized sources or standardized methodologies or standardized phantoms that could be engaged and knowledge leveraged for this standards development activity. So I don't want to be. I'm very mindful of being myopic with regard to what I know from the US, but don't know of everything international. So I would encourage people to let us know kind of who should be engaged in this standards development. And I would say beyond standards development, actually, how should I say, standards verification. Because some of these things, I think getting the reproducibility process and understanding, as we talked today, about bias and variance in these measurements is critical. Like Stephen showed us, being able to say, I have a standard methodology, this is the variance in that standard methodology, where does your method compare and how does it compare? Because as you saw in some of his slides, moving from vendor to vendor, you can see already that there's some variation. And so being able to drive that variation down and shifting any specific bias that they might have is very, very useful for the field.
Yeah, that's a great. We'll include this in maybe in the working group related to documents and existing standards. Is there anyone who wants to comment further on the topic of standard development? No. So let's move then to the last item that we had on our list, which was about the resources. So what do we need to conduct this further works and developments in terms of, do we need additional expertise? Do we need logistics? Do we need fundings? I maybe can say just a word from the foundation perspective that I think we will be able to provide logistics support, for example, so we discussed a data repository repository of software and things like that. So Eleanor suggested GitHub, of course. That's a good way to do it. We have also a data repository that is curated by something in Charlottesville called the Open Science Foundation that we could also leverage to enable sharing of data sets and again, links to GitHub and documents in terms of methods and protocols. And we should be able also to support logistically the organization of these work groups and future discussions. So yeah, so it's open to again to discussion about where do you need are the needs in terms of additional resources to move on. Yes, Constantine.
So GitHub and all the things you just said, Frederic, are a great thought. I think where the foundation specifically could really help is, you know, what tends to happen to these repositories in the digital world is they go through a period of glory, usually in the 3 to 6 months after all this activity has happened. And then they cease to be maintained and they cease to be updated and they cease to be current, which is why the effort doesn't evolve. I don't know if there is a person within the foundation who's perhaps a standards officer or so it would ideally need to be someone with enough scientific background to be able to navigate what's in that section. But it would be great if we could create a portal that is live and receives updates from users and someone maintains it. And as we start creating publications that refer to the standards, they appear on that portal. And as we start creating standards, they appear on that portal so we can grow and maintain it as a resource. I think the foundation with its resourcing could play a really crucial role in not only creating the IT side, but also maintaining the library and evolving it. And then from a other support perspective, you know, we had a discussion with Sonic Concepts and potentially other manufacturers early on in the workshop about creating some prototypes and some initial ideas for these calibration sources. And then Brian frequently referred to the term round robin. Well, maybe development of that prototype, if needed, I don't know if funding really is needed, of the associated resources and of the subsequent round robin experimentation from lab to lab could also be a foundation supported project that involves many of the people currently appearing in the right-hand column of this chat.
Yeah, so Constantine, that's a very valid point. We have found folks like Sam Howard and the industry folks to be very supportive of the idea of creating prototype phantoms for doing these types of round robin evaluations. So, you know, various companies that build phantoms have been willing to build prototype phantoms in order to do testing. Some fairly sophisticated even for being able to do that type of testing and proof of principle, particularly in the Kiba activities. We've even seen companies willing to provide some resources in order to cover the costs of moving these phantoms around from place to place to be able to do those evaluations. What's been, you know, somewhat missing is any, you know, large scale, monetary support. Obviously, we've been very fortunate at Kiba to have some very willing volunteers and very interested scientists involved in those activities. But ultimately, some of these activities begin to get larger scale, in which case we are going to have to look for support from various funding agencies in order to be able to support that. And nothing speaks better to that than having, as you might say, preliminary data, right? that says that this activity is important and it needs to be done. And these are all the reasons why. And this is what we've done in this area already, to be able to drive the interest in additional resources to help facilitate further development. And obviously, this is in the interests of the therapeutic ultrasound companies as well, because it provides a mechanism for being able to create some level of standardization and improve their understanding of what it is that's being measured in their given systems. I think this is the right time to be looking at this problem, because it isn't that everything has already been done such that you're having to re-engineer things necessarily. One thing that was mentioned during the course of the workshop is when you look around the room, and obviously I can't look around the entire room, and I don't know everybody's expertise, but in some cases, it was mentioned that what we're missing basically is the biological side of this. In other words, the understanding of the downstream effects of the physical interactions that cavitation or these bubble activities have. So is that an area of expertise that we're going to have to draw in? Does it seem to the group to be at some level conspicuously absent? So does that comment still hold?
So I think Brian, that we specifically also discussed the, for example, the effect of microbubble on the vasculature, or when we were talking, for example, about thrombolytic treatments and things like that. So that might be an area where expertise is still needed, I think.
Yeah, we didn't have, a workshop session that was really kind of devoted to that type of physiology and biology and so on and so forth. And so that may be a gap that we might need to fill. So if you circle back to that slide, your slide number 2, that's in here, most of what's in this slide were the actual topics of our workshop. As you can see from items, the first 4 items that are in there, but we had less about the bioeffects and interactions and therapeutic endpoints, and certainly not as much about the clinical outcomes. So that may be kind of something to consider for our future activities as well, to maybe think about a deeper dive on that aspect. Kostas?
Yeah, I think we might also end up kind of like, in the direction that we end up today, that we felt like we do have enough information, we do have enough knowledge, but we can certainly learn from what people have done in other fields. So I think going down to ultrasound mechanobiology, it will be a necessary step for us to be in position to somehow quantify the forces related. Because if somebody comes into the field, we'll say, okay, what do we do to the cell, right? So the first question is, how can I relate with the forces that the cell is able to sense with what are the forces that you are, the bubble dynamics potentially might radiate. So I think that it will also need to use that workshop maybe as a stepping stone to move closer to be in position to come up with what these acoustic emissions mean in terms of forces, and then people that have already studied these forces in a different context will be able to really facilitate this conversation moving forward.
That's a very good point, that to the level that we can understand what our mechanisms are, then we can start to ask people who are involved in the biology of similar mechanisms What should we be expecting to see? What are the downstream consequences of those activities? That does make a lot of sense to me.
So no further comments. Maybe it's time to conclude.
Yeah, I think so. Any final comments from anyone in the group in terms of what you've heard today? Well, I certainly want to thank everyone's participation. We were as many as over 50 people involved in the conversation today at various points. And I think, I don't know what the final numbers were. I know at one point we had over 300 registrants for the workshop with different attendance levels at different points in time. Certainly my thanks to Emily for helping with the workshop and doing so much, Jason and the AAV tech team for helping us. Fred, any final comments?
No. Just I will associate with myself to the acknowledgements to Emily, Jason, and everybody who participated in the workshop. It's been a great adventure.
Yeah, special thanks to the moderators, the speakers, the organizing committee, all of the people who are participating.
Yes, Costas, you want some final word?
I'm just like say thumbs up, no, don't say something.
Oh, okay,
Thanks to everybody, and I think it's remarkable that we reached to a point that we have so much agreement about so many different topics and important topics.
Constantine, were you? Yeah, go ahead.
I was just waving, saying a big thank you to everyone. That's all. and about to go and have a Friday night in the UK. The first Friday without a cavitation workshop next week. How will we ever survive?
We'll take a little break, but we can't lose momentum.
Happy Halloween, everyone.
Thanks, everybody, and thank you for your attendance today.
Thanks, everyone. Bye.
Have a great weekend.